Okay, hello everybody. So today I figured we can go and start talking about the next tool that we were using after FastQC and the multi-QC to summarize the FastQC results, and that is Trimmomatic. So this is actually something that we've already started running in the video Bioinformatics, SRA, download, QC, and trimming. And today I wanted to talk a little bit about what the flags that we're inputting into Trimmomatic actually mean. So instead of just blindly running the tool, like copying, pasting, what I've noticed some people do when they're going through tutorials and not really understanding what they're doing, they're just copying and pasting. But we can go through and learn a little bit about what actually is happening with Trimmomatic when we're running it. So first off, I'll talk a little bit about some of the other tools that are available for trimming. When I first started my bioinformatics learning and doing it at uni, I guess, I did btrim. That was a tool that was recommended to me by the bioinformatics professor. And btrim is kind of old, it's from 2011. But I haven't heard anything negative about it, and I think it's just something that people have moved on now to Trimmomatic. And also we'll talk a little bit about another one that we might look at in the future too. It's a little bit newer than Trimmomatic. Another one is Skewer, and what Skewer does is it actually just looks for the adapters and removes them. I don't think it does any actual quality trimming, just the adapter trimming, which is really valuable too. And we'll talk about that a little bit later, why the adapter trimming is important. And I'll put all of these links down below if you want to go through and read the abstract or the paper. I think one of them is not open access, but then there's FastP. So this is the one that I was talking about initially that we might go through and do a little bit later. FastP, I just want to read the results because they sound really interesting. This is from 2018. The results say that they developed FastP as an ultra-fast FASTQ preprocessor with useful quality control and data filtering features. It can perform quality control, adapter trimming, quality filtering, per read quality pruning, and many other operations with a single scan of the FASTQ data. This tool is developed in C++, which is valuable, and has multi-threading support, also valuable. Based on our evaluation, FastP is 2 to 5 times faster than other FASTQ pre-processing tools, such as Trimmomatic and CutAdapt, despite performing far more operations than similar tools. So I'll just go back to the C++. Programs that are written in more of those base languages don't have so much overhead, so a lot of tools for bioinformatics are written in C and C++, whereas if you go into R and you load a library, and all of a sudden you have all of this library overhead. They're loaded, but you're not necessarily going to do anything with it. So the RAM for R can just get crazy high even if you're not doing anything and you remove all of your files. But yeah, so this is something that I might want to take a look at, and maybe I'll make a video on it in the future too. It sounds like it is possibly a really good tool. There's also a GitHub link that I'll post below. And then moving on to the topic for today, Trimmomatic, which we had made use before. This is from 2014, so it's a little bit newer than the btrim that I was using, but it's not as new as the FastP. So I'll talk a little bit about how I got to using Trimmomatic. Like I had said, when I first started, I used btrim in 2 RNA-seq projects, and I tried using it in a DNA-seq, and I couldn't find a binary that worked. I don't know if I just wasn't looking in the right places or not, but and then I went on to do a workshop for bacterial genome sequencing. So we provided the DNA for some bacteria that we thought might be novel, They had really low 16S sequence scores when we sent them in. And so we sent them in to get whole genome sequence, and I was part of that group. And we started using Trimmomatic, and that was reassured when I went on to do another course later on that did RNA, microbiome, and some genome sequence work too. And they were using Trimmomatic in that, and it was just another reaffirmation that this is a good tool. So we'll get right into it. So the first thing that we specified when we were working with our Trimmomatic, the first flag, was we had to say whether or not our reads were paired end or single end. So first we'll explain what the adapters and the indices are. So the common adapter, I just called it the common adapter. I think you can just refer to it as adapter, but this sequence at the very, very end of what would be your read is actually what's binding to the flow cell. So when you load your DNA into the sequencer, this very, very end part is what's binding to the cell. This next part that I called the sample unique index, each sample is going to get a unique index for it. So then you can tell your replication 1 from your replication 2, and so on and so forth. So each sample has a unique index, and that comes after the adapter. So the adapter is a hard bind, and then your index is actually red. So that's going to be on your forward read, and then you have your reverse read on the bottom. And, well, you have your reverse strand. And for single end reads, what happens is this binds, and then you just read through. It's just one way thing. But for paired end, if you look at some of the images that Illumina has for describing their paired end sequencing, they have the common adapter, and then they have their index, and then the read in the middle, and then they have their other indexes and the adapter on the other end. So what happens when they get done reading through the sequence one direction, they'll flip it over, and then they'll sequence the complement or the other direction through. And so that's how they get their paired end reads. I have this here to show how paired end reads can be more beneficial than single end reads, but this is just a single end depiction. For single end reads, sometimes you'll see the quality at the very end drops down really far, so you can have the end of the read have bad quality, which kind of stinks. And paired end reads, you generally have a little bit better quality. It's good for transcriptome stuff, and when you need high read counts. It's also really good for looking in those transcripts for novel splicing variants and also some insertions and deletions. Paired end reads with the increase in quality is also really beneficial for some genomes. If you're trying to do a de novo assembly of genomes and you don't have that template or the reference genome, paired end can give you higher quality for that as well. So for single end reads, it's not going. This is probably not the right way to think about it, but for quality for single end reads, I think of it as, or when you have really long reads, I think of it as like the inflatable man. When he's flopping around, you have more unknown towards the ends, and as it gets closer to the chip, the quality is then better than really far away. So the shorter reads, you can have better quality. So what we're working with, we have 51 base pairs, so it doesn't really have a whole lot of wiggling around, I guess, but that's another thing to maybe consider. The next thing that we were working with is the IlluminaClip. Well, so We did the paired end and then threads, so this is kind of pretty straightforward. We gave it 5 threads because it didn't go beyond that, and then we had all of our inputs, and then we gave it the base out. So the base out is going to tell it what to give the name of all the outputs. So we have trimmed reads, and then the squiggly brackets. So the squiggly brackets just mean whatever is being fed into it, like I mentioned last time. So everything is going to go into the trim reads folder with the same SRR blah 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 number before it gives it the 1 paired, 1 unpaired, 2 paired, and so forth. But then we have IlluminaClip. And for IlluminaClip, this is where it's actually going to look for our adapters. So we have our adapter list, and this is what comes when you download Trimmomatic through the Conda, and it might be different if you do the installation a different way. I'm not sure. I just did the Conda. But so we have the first section, so we have IlluminaClip and then colon, and then the first thing after that is going to be the name or the file path to our read file. So we moved our read, our adapter file to our working directory that we were in. That way we could just give it this name. We didn't have to type out the path all the way to where all of these were. The second number is going to be the max number of bases that can be mismatched in order for Trimmomatic to still recognize that sequence as an adapter. So we gave it a 2. The next number is a score, so it's an alignment score of 2 reads that have adapter contamination. So we have 30. The higher this, the more stringent. For 30, I think somewhere said that means 50 base pairs long have to match with the adapter contamination. The 10 is going to be the score given to adapters that match when in the read sequence. So kind of like this, the longer this, the more stringent. 2 here is the last 2 specifies the minimum adapter length in a palindrome mode. I'm not exactly sure what that means, but they keep both reads. So the default for this 2 is actually 8, and I just put 2 there because that's what's recommended by the Trimmomatic page. But to keep both reads is something that's really important. When we feed in our files here, we have a forward read and we have a reverse read. When we then go to align our output from Trimmomatic to our genomes, and with our GTF file using star later, we're going to want to have paired end reads to feed into star. If we didn't keep both reads here, what we would have is sometimes a read would be redundant, and when a read becomes redundant, it's going to throw one of them away. Well, if it's going to throw one of them away, we no longer have a paired end. We just have a single end, and then that can cause some problems. Or we could throw away too many, since we have 4 output files. If they're going into one of the unpaired files, we could end up with a higher unpaired than is actually not paired. So that's really important. There are ways to go around it, but the end goal of this project is to make a workflow to where you can just feed in your raw reads and it does everything. And with SnakeMake, it's way easier if we just keep both reads, because then we can say all of them have a forward and reverse when we start mapping against the genome. Next, so this is leading, and we also have trailing. They're pretty much the same thing, but these 2 flags cut off the bases, the beginning of the end or the end of the read. So in this case, it's the beginning. So we start here and we're going to move to the right, or we're going to start here and we're going to move to the left. So leading is at the beginning of the read, and trailing, if we start here, go to the end, this is the end of the read before then we would have the next index and the adapter. It's going to figure out whether or not this quality is below, and here we specified 3 for our quality. If it's below 3, it removes those base calls or those base pairs, which is pretty straightforward. Next, we have min length, and this comes into play when your read length is really short, and then you start, if you had bad quality, so if we go up to here, we have really good quality from or through everything, but if we had really bad quality, well, I guess it's on this page too. If we had really bad quality, then all of a sudden we would start throwing away more of our read than we would be comfortable with. So here we're saying if we're starting to throw away too much of our read, our read that we started off with 51 base pairs, if we drop below 36 base pairs, we just have to discard that read. We're losing too much information, and that read can start causing more issues or not be as helpful or compute worthy to continue using if it gets below a certain length. So we specified 36. If we lose 15 base pairs, just get rid of it. And that really comes into play when we look at something like this, so a sliding window. We have really good reads, so it's not too big of an issue, but when we start getting longer and we have a really hard tank at the end of our quality, we're going to say in a sliding window of 4 base pairs, so from 1 to 4, if that average quality drops below 20, just cut it off. So for instance, here's below 20, here's below 20, below 20, below 20. So probably for read 10 or a base call 10, the average of 10, 9, 8, and 7 is below 20. We would cut that read off. So that read is now only going to be 10 base pairs long if the leading didn't get rid of any base pairs. We have 10 base pairs long. Well, that's way below 36, and a read length of 10 isn't really going to be that helpful, and so we can just get rid of it. And this quality score at the very end really depends on what you're doing. For RNA-seq, I've seen people drop it down to 20 or not even do sliding window. In our case, we didn't even do it because our read quality is pretty good across the board for everything. But for DNA, where you want to build a genome, 25, 27, even 30, getting that quality really high, that way you're very confident and you're keeping only the absolute best quality reads to build your de novo genome. And then finally, I did it the hard way. Just reading through the manual for Trimmomatic might have saved this issue, but there's actually a trim log flag. And that trim log flag would go, if I can get that out of the way, would go here after threads or before threads. So we have Trimmomatic paired end, we're going to give it 5 threads, and then we would tell it, put our log in this file. Instead of taking the standard output and feeding it into a log file, we could use this flag. And that is just a lot more neat, but we still have to use that cat pull. So we have to pull in whatever we're outputting from the cat in our SRR accession list to be input in here. It's not going to pull from our input names. We'll have to do that manually. So that's a little bit what Trimmomatic is doing in the background, or what we're telling Trimmomatic to do with all of the inputs that we're giving it. Later on, maybe we'll go back through. This is really interesting to me, the FastP, and that's from 2018. Where did I see that? Yeah, 2018, whereas Trimmomatic is from 2014. So maybe this could be the next up and coming thing. From there, we can go over to Ubuntu window. So we're actually in our Ubuntu window now, and if we do ls, we'll get a list of everything in here. You can see that I already have our multi-Q output, multi-QC output, but we can remove those. We'll just remove multi. It's a directory. It's a directory. Oh, it's a folder. That's okay. Anyway, so we're in this folder. We can see we have all of these logs, and I'll just go through and delete it over here then if it's not going to like that. Let's see, that work? Yeah, so now we can do multi-QC and then dot. So we're going to tell multi-QC, look through this folder, see what you can find for log files, and it knows Trimmomatic log files. So you can see here, it found 12 log files, and it now put our report in a multi-QC report. So if we go to Firefox, we can see this is the output multi-QC gives us. Cool. For our Trimmomatic stats. And it looks like overall a large chunk of our reads were kept. We can actually see that down here too. So our surviving reads stayed pretty high. Generally, I've heard people say between 20 and 50,000 or 50 million reads is good for paired end, but we have at least 55 million and up. So that's pretty cool. So we have our surviving reads in blue, and then our forward surviving. So of the _1 reads, only those that were kept. And then in the little bit darker purple or darker pink, we have only the reverse reads, the _2. So where there was a pair between forward and reverse, where only the second one was good enough to keep. That's going to be our dark pink. And then our deep red is going to be those that were dropped out. So here we had 16% of our reads dropped out. But overall, it looks like our commands for Trimmomatic worked out pretty well. And here we have the lowest amount kept was about 70% of our forward and reverse paired, which is going to be the surviving. And that's what we'll then feed into star. So yeah, looks like everything is good. Next video, we can either talk about star, what star is doing when we give it commands, or we can go ahead and run star and then worry about that later. So see you guys next time.